Hello, everyone. Thank you for joining us today to Tomorrow Health, the Future of Cancer Immunotherapy. My name is Sophie Cherney, and I am Switzerland Innovation Commissioner to the United States. Switzerland Innovation was initiated by a public-private partnership to create a platform where research groups from Swiss leading universities and hospitals on one hand, and innovative worldwide company on the other hand, can meet, can exchange, and can launch joint projects. Next slide, please. Today, Switzerland Innovation Networks unite 6 innovation parks across Switzerland, which is more than 30 academic partners, hospitals, and teaching hospitals. I am really delighted today to host 6 wonderful minds and bright minds from Switzerland for an enlightenment discussion. And following those 40 minutes of discussion, we will have 10 minutes breakout session. You can see on the left side of your screen, there is thematic breakout session. There's 3 of them. At the end of the discussion, we will open those rooms. You can choose the room of your choice, and then obviously you can interact with our speakers. With no further ado, I would like to introduce the next speaker, Dr. Brian Hashemi, who is the co-founder of Converge in Oncology Summit. Brian, the floor is yours. Thank you.
Great. Thank you very much, Sophie. Hello, everyone, and welcome to the panel on the future of immunotherapy. We're delighted to share with you just a glimpse of what is happening in the innovation landscape in Switzerland. As you may know, Switzerland traditionally has been very strong in innovation within the healthcare sector for many decades, and there are a number of excellent academic centers in all the 3 major regions of Switzerland, whether it's Swiss-German part or Swiss Romand or the Ticino area. And we have speakers today represented from each of these regions, and both from the academic centers of excellence, which then lead to spin out and scale up companies. We'll have 2 of those represented here to share with you a little bit of how the innovation landscape is nurtured, how an idea from a benchtop in the academic center ends up into a company. We have 2 examples of those companies that have actually scaled. One was acquired through an M&A by Roche that will be presenting their story, and one which scaled and finally did an IPO on the Swiss exchange, which will share with you that story. So we hope to give you a little bit of a glimpse on how innovation is nurtured and cultured and supported by the investment community to go from an idea into a startup, into a scale-up, and finally into a mature global player making an impact in immunotherapy. We try and keep our discussion focused around patients and patient benefits, and we hope that you will enjoy the discussion, and we'll look forward to seeing you in the breakout sessions for the Q&A after the presentation. Great. Without further ado, we're going to start with our first speaker, Dr. Pablo Umana, and I look forward to hearing your view on the innovation landscape and sharing your story about what you're doing in Zurich. Pablo, the floor is yours.
Thank you, Brian, for the nice introduction. Could we go ahead to the next slide, please? Yeah, my name is Pablo Umana, and I head the Cancer Immunotherapy Drug Discovery Group within the Roche Pharma Research and Early Development Organization, and I'm based in the town of Schlieren in the canton of Zurich in Switzerland. I will just tell you a little bit about our case. As Brian mentioned, we're starting from as a spin-off from the ETH in Zurich, and then becoming part of Roche Pharma Research. And in the context of that, also what is the work that we have been doing in the field of cancer immunotherapy and innovation in that area. So if you go to the next slide, please.
Pablo, are you still there?
Yes, somehow the slides are not moving for me, but it's okay.
Okay, you are on the next slide, yes.
All right, okay. Okay, thank you, there is a delay. Yeah. So our research site nowadays is called as part of the Roche Pharma Research and Early Development Organization, or PIRET, part of Roche Pharma. Is called the Roche Innovation Center in Zurich. But originally we started, the research at the site started as a spin-off from the Swiss Federal Institute of Technology in Zurich, or the ETH Zurich. We were working there, developing a new technology on how to engineer anti-cancer antibodies to engage a certain component of the innate immune system into attacking the cancer cells. And based on that technology, we spun off a company still staying in Zurich. We generated new anti-cancer antibody drug candidates. And the first one of those was a molecule that today is known as Gazyva, or obinutuzumab. It's an anti-CD20 engineered antibody, which was then the main driver for the acquisition of our spin-off company 4 years later after foundation by Roche Pharma. And at that time, we were 29 FTEs, 29 employees. We became integrated into the research organization of Roche, both in the oncology research, but also in the field of protein engineering. But we remain in our site there, and over the years, this site has now grown from 29 people where we were in 2005 when we were integrated. We are now approximately 200 researchers working still in the field of cancer immunotherapy. It's just that we have brought up and developed new platforms for engaging the immune system to attack cancer, and we have become a research center of excellence within Roche for cancer immunotherapy. We have a new building dedicated to our research site, which you can see there in the picture. Some of the achievements that we have had together with the rest of the Roche group, of course, have been, well, first, the molecule that was the driver for the acquisition of our biotech company, Gazyva, that became the first ever therapy approved by the FDA under the breakthrough therapy regime. It is used in frontline follicular lymphoma and chronic lymphocytic leukemia, and currently is in phase 3 trials for lupus nephritis and autoimmune diseases. Then the next molecule from our T-cell-based cancer immunotherapy efforts is glofitamab, which is currently in phase 3 trials. And we have additional significant number of molecules, over 10 molecules in phase 1 clinical trials. These are different types of cancer immunotherapies. Next slide, please. So just to give you a flavor of the type of cancer immunotherapy work we're doing today, we're working on new ways to both engage and redirect the immune system to attack cancer, in particular T cells. And then once we have that attack ongoing, we want to strengthen it by adding other immune modulators. One of the approaches of the platforms that we have generated within Roche are these T-cell engaging bispecific antibodies. We have worked on a special format that is shown here that we call 2-to-1 format. I don't have time to go into the details, but basically we have been working on optimizing both the potency and the therapeutic index generated by this type of molecules. The most advanced candidate is the one against CD20 that, as I mentioned, is already in phase 3 clinical trials in the Roche group. But we have various others at different stages. We have against both solid tumors and hematological tumors. We have some that actually mimic TCR, so they are directed against intracellular proteins by means of targeting peptides from those proteins that are presented on MHC molecules on the surface. And we have also presented recently on the next generation technology where the molecules are activated by tumor microenvironment elements to further enhance the therapeutic index. Next slide. And just very quickly, an example of that is as I mentioned, the glofitamab molecule, the CD20 antibody, which is now in phase 3 clinical trials. Recently, we've presented just a few days ago at ASCO, we gave an update of the clinical data that we are seeing with this molecule, which I think is really impressive. This is relapsed refractory patients, third line plus patients, both in aggressive and indolent lymphoma. And we are seeing complete response rates of over 70% in both indications. So this is really on par or even superior to second generation CAR T cells. But of course, with a molecule that has a very manageable CRS, cytokine release syndrome profile, and no significant neurotoxicity, and of course is off the shelf. Next slide, but we want to keep building on this. And for this, as I said, we are adding a next generation of compounds. Once you have the T cells redirected and engaged, we want to further strengthen those T cells by providing tumor-targeted co-stimulation. And we have then also pioneered in the field the combination of off-the-shelf T cell bispecifics with second-generation tumor-targeted co-stimulatory agonists, in particular of the 4-1BB co-stimulatory receptor. We have two such combinations in the clinic now, one for hematological tumors with the CD20, CD3 bispecific combined with the CD19 4-1BB agonist, and the second one is our CEA CD3 bispecific for colorectal cancer combined with a tumorous trauma antigen targeted, FAP targeted 4-1BB agonist. So these are really first of a kind combinations in the whole industry for this type of approach that have reached the clinical stage. And the next slide is just an example of the preclinical data that we have achieved with the CD20, CD3 combined with the tumor-targeted co-stimulation. As you can see, sorry, can you go again forward? As you can see, we have much significant improved efficacy, and as a result of a much increased let's say, fitness and accumulation of engaged T cells in the tumor microenvironment. So this is analogous to what second generation CAR T cells did, but of course here we are doing it by combining two off-the-shelf reagents so that we can reach the majority of patients worldwide, we hope. And our dream, of course, is to, by bringing this kind of activity, that we in the future may replace chemotherapy with this type of targeted therapies. And next slide, I think.
Great.
That's it.
Thank you very much, Pablo, for the first presentation on combination therapies. When we talk about immunotherapy and trying to maximize the response in the patients, so we have two speakers on combination therapy, and our next speaker, Andrea Cerdeira from Molecular Partners, will share with us how they are following combination therapies with the DARPin platform. Anna, the floor is yours.
Thank you, Brian. Maybe we move to the next slide. So my name is Anna Cerdeira, and I'm heading the strategic planning and portfolio management of Molecular Partners. And we are literally neighbors of Pablo. So we are also located in Schlieren. But instead of working on monoclonal antibodies, we work on a protein scaffold, the DARPin platform. So Molecular Partners is also a spin-out from University of Zurich, but it has a different story from the one of Glycard. So we are a Swiss biotech with currently 150 employees. And as I mentioned, the company was founded in 2004 as a spin-off from the University of Zurich based on the DARPin platform. So far, we have developed product candidates across 3 different therapeutic areas: oncology, virology, and ophthalmology. And we have also established major partnerships across these therapeutic areas. Our first partnership was with Allergan, that then became AbbVie in ophthalmology. Then we also partnered our Fab4-1BB with Amgen. And most recently, we have Novartis as a partner for Ensovibep, our COVID-19 molecule. We have currently 2 programs in clinical, one in phase 1, the Fab4-1BB, and one in phase 3, Ensovibep for COVID-19. So next slide, please. So how do we go from a university spin out to a company listed in the stock exchange market? So it all started in 2004, where a group of Ph.D. students founded a company, Molecular Partners, and they started with seeding money. And they use this seeding money to develop the DARPin platform, as well as to generate data on the initial candidates that was a beekeeper that could attract venture capital investment. And this happened in a Series A and a Series B. Until 2014, the company had attractive data enough with the first clinical candidate, and the company decided to go for the SIX exchange, so where it became publicly listed. More recently, now in 2021, and I will skip 2020, we have announced our intention also to be listed in the US listing. So, and the goal is to become dual listed in the SIX exchange as well as in NASDAQ. Our partnerships have evolved from really service, more service provider to capability deals, because where we are true partners and we have development capabilities, because we have evolved as a company really as well as in capabilities. So in the next slide. So what are DARPins? So DARPins, as I had mentioned, is a protein scaffold. So if you have a monoclonal antibody on the left side for comparison purposes, so the first thing you can see is that DARPins are smaller. So they are 10 times smaller than monoclonal antibodies. They have a single monodomain binding region, and they don't have an FC region. So this means that we need to use human serum albumin to modulate the half-life. These are our manufacturing can be through microbial expression, and we generate stable molecules. And we have demonstrated that DARPins have a clinically safe profile. Next slide, please. So now I bring two examples of the application of the DARPins in immuno-oncology, the first one in solid tumors and the second one in liquid tumors. So the first molecule here is MP317, that is a FAP-CD40. And you will see that in both examples that I bring, I want to highlight one of the main advantages of the DARPins, that is the ability of forming multi-specific molecules with multiple targets. And MP317, you see that there are two binders targeting CD40, one FAP and another one for to link to human serum albumin to extend half-life. And here the objective is, because CD40 is a very interesting target, but it is known to have those limiting toxicities through systemic toxicity. So here the objective is to use FAP that is present in large quantities in many solid tumors, but not so much in healthy tissues. And the objective is to utilize FAP to cross-link and only then have the CD40 activation. And like this, we have a local action with the activation of antigen-presenting cells and activation of T cells without systemic toxicities. So in the next slide. So in the next slide, we bring here an example of what DARPins can do in liquid tumors, also using multispecificity. And here specifically, we bring an example of one of our T-cell engagers. This is not the final product candidate, but it's one of the product candidates that we are testing. And in this case, it's not very clear in the image. We have a CD3 binder, and then we have binders against 3 targets of interest in AML. But we know from the competition that these targets are not clean targets. This means that we have many times of target toxicities and hematological issues. And in AML, it's very important that we kill the malignant cells, so the leukemic stem cells and also the blasts, but that we leave the hematopoietic stem cells and the progenitor cells. And so how do we achieve that? What is our hypothesis? So we we develop DARPins that have an engineered affinity. And what we want is to, with these DARPins, when we just have one target, we have low affinity binding, and therefore we don't have any activity. But when we are in presence of two or more of these tumor-associated antigen, through avidity gain, we have higher affinity, and therefore we have an activation of the immune system. And by doing that, we manage to increase the therapeutic window. And our hypothesis is, by doing this, that in patients we will achieve higher efficacy, more durable responses. We will also be able to target the heterogeneity of the tumor by targeting multiple tumor-associated antigens, and also reduce the off-site tissue toxicities. So thank you very much. I hope you enjoyed the next talks.
Thank you very much, Anna. That was a very nice presentation of another combination therapy strategy. And for those of you who are interested in combination therapy, after the panel is completed, don't forget you can join both Anna and Pablo in the combination immunotherapy breakout session for Q&A. We'll now move to the academic centers of excellence. We'll start with Professor Lana Candelaft, who's a leading scientist here in the Canton of Vaud area, particularly in T-cell therapies, and the floor is yours, Lana.
Thank you. Next slide, please. So I would just like to take you back to basics, because I will talk more here about cellular immunotherapy. So if you see,
Lana, you unmute. Can you please restart? There you go. Please restart on the slide. No problem.
I'll take you back to basics, to cellular immunotherapy, because what I will be discussing here is mainly how we built a program to take cellular immunotherapy to patients. And so here's just a slide to show how we have two major players, which are the dendritic cells and the T cells. The dendritic cells are the cells which pick up antigen, take it to the lymph nodes, where then they activate the T cells, where we think T cells are soldiers which can attack the tumor. And so here what we can do is either reinvent or mimic this cycle, reinvent this cycle either by vaccination, which is based on dendritic cells, or basically targeting these dendritic cells, or by adoptive T-cell therapy, which is what I will show you has been done in Lausanne after building the infrastructures. Next slide, please. So as we all know, the steps to take a concept from bench to bedside are numerous. It seems here very small in this slide, but however, the amount of work it takes specifically to take a basic preclinical product to a phase 1 or phase 0 proof of concept product is a huge amount of work and requires an infrastructure. And so when we came here to Lausanne in 2013, this is what we basically started building this infrastructure that takes therapies from bench to bedside, and also understand what's happening in a phase 1 clinical study and basically take it back to the lab in order for us to understand and improve the therapies further. Next slide, please. So as you know, these are really two death valleys. The first is taking this concept from a university setting, from a bench, to what we call GMP adaptation. And here's when in cellular therapy, these products are not just a simple pill, they are really biological products, alive products that should be produced and manufactured in a GMP environment. And here, if we don't have the infrastructure in that bridge, we have the first valley of death, and the second is really taking this cellular therapies to patients and implementing it in a clinical setting, which I'm sure is known now that the CAR T cells are out and given to patients. I'm sure everyone has faced many challenges. So this is what we focused on in Lausanne. Next slide, please. And therefore, what was my job to do here is to really build a structure. So is a center of experimental therapeutics. It's a service within the Department of Oncology that is really the bridge. It's a structure of about 140 people, and at the moment they're nearly 160, which are bringing this innovation from lab to patients. So thanks to the investments which the Canton Vaud has put on the table, we were able to build that structure with different teams, clinical platforms, manufacturing platforms, and translational platforms in order to solely do phase 1 studies for our patients. Next slide, please. So this is a little bit animated, so it's difficult just to show what we have done. So we have really submitted, we are running submitted 200 clinical trials since 2016. We are running about 108 clinical studies. We have about 600 patients in clinical studies, and 12 studies were our own studies. So concepts developed in Lausanne and basically taken with all the regulatory authorities. It's our own products that we are developing that then are taken to patients. Next. So out of these were basically two vaccine studies and 2 adoptive T-cell therapy studies, and multiple others which are dealing with combination immunotherapy. We also kind of set the stage of how there are so many challenges, yet advantages of manufacturing in a university hospital setting. So it's a difficult job to achieve because it's completely different than having a manufacturing system or a manufacturing facility that is just purely manufacturing. Here we are really trying to impose and implement this and force it into the normal hospital setting. So we have set the research laboratories, a GMP facility, and the clinical way of actually dealing with these aspects. So just a couple of slides, next please. So here we established our own cellular manufacturing facilities, which has the capacity of producing about 300 products per year. It can produce genetically engineered and non-genetically engineered products. Next slide, please. We have also established a phase 1 unit dedicated for clinical research. So it's an inpatient unit that has the capacity to treat about 170 patients per year. Next step, please. And also an outpatient unit completely dedicated for clinical research. So it's really a floor that all we do is phase 1 studies, and what we dedicate all the staff is to do this kind of work here. Next slide, please. So the first really study we embarked on was doing adoptive T-cell therapy of natural tumor infiltrating lymphocytes. So what is that? Basically, we can from the tumor itself, we can isolate these T-cells, these soldiers. And as you can see here on the left-hand side is basically the tumor is chopped. And this is a picture from our own lab, chopped into small pieces. Then we undergo what we call a pre-rep, where we now expand these T-cells that have been isolated to the tumors, and they are put in these little G-Rex. And then after we have the TIL expansion, which is the rep, and you can see these big bags that basically multiply these T-cells to billions. And then basically we do the infusion to the patient in this bag that is been shown on the picture. Next slide, please. So we have opened 2 studies. This is just to show you one that has been completed in melanoma. Here is Philippe doing it in the lab, showing please the last slide, and then another slide. And this is, no, please go back. Yeah. So this is the first TIL product that has been infused, 50 billion cells. If we play that video, we show that these T-cells, can you try to play it, Brian, please? Thank you. So we show that these T-cells that we have grown can actually kill tumor cells that are taken from the same patients. So not only that these T-cells have grown to billions, but they're also specific and can target and can kill the tumor cells of that specific patient. Next slide, please. And here's an example in Lausanne of how a patient with late disease melanoma has been treated with this TIL product. And as you can see here on the scan, how basically all the lesions have disappeared 18 months later. So this is one of our first phase 1 proof of concept studies that have been done. Next slide, please. So here, just to show that we do have an infrastructure and expertise to allow the transfer from basically bench to bedside. Next slide, please. And then, yes, and then I had more, but then this will be what we are building more, and the next generation of T cells and the vaccines could be discussed in our breakout session. Thank you very much.
Great. Thank you very much, Lana. And now we go to the next Center of Excellence in academia, and we go to Dr. Professor Andrea, and the floor is yours.
Hi, everyone. So my name is Andrea Alimonti. I'm a full professor of experimental oncology at della in Switzerland. However, my lab clinical activity is located in Ticino, which is in the south part of Switzerland. Next slide, please. So my lab and clinical team work around the characterization of the tumor microenvironment of prostate cancer. We believe we want to try to develop novel therapy for the cure of metastatic castration-resistant prostate cancer that, as you know, is still a type of cancer which is uncurable. So there are very few options for this patient, and is one of the major causes of death for cancer in the West world, of course, in men. So many years ago, we were very lucky to discover that the tumor microenvironment of prostate cancer patient is characterized by a strong infiltration of a subtype of immune cell that we have described for the first time in 2014 in this Nature paper, that are named now myeloid-derived suppressor cell. This has opened a new field of investigation. As you can see, there are now many papers that have validated the presence of this myeloid-derived suppressor cell in prostate tumors. And as you can see also from this slide, there are additional immunosuppressive immune subsets such as tumor-associated macrophage or B cell, which can render particularly complicated to use a modern immune checkpoint inhibitor therapy for this class, for this type of patient. Next slide. This is a slide that shows what we actually do in our lab, and later on I will show you what we do in the clinic. Our discoveries start from mouse models of prostate cancer, which are characterized by loss of this tumor suppressor gene that is called P10. As you can see here in this slide, the tumor microenvironment of this prostate cancer mice is characterized by the presence of these myeloid-derived suppressor cell, the tumor-associated macrophage, that are really the major component of the tumor microenvironment of this prostate tumor. However, if we even act therapeutically in this mouse model, we try to suppress the androgen signaling by treating the mice with androgen deprivation therapy that are standard of therapy for patients affected by prostate cancer. There is a further expansion of this myeloid-derived suppressor cell in the tumor microenvironment of these mice. Next slide. So if we try to observe whether, can you also, there is another animation, can you click? So one slide back, thank you. So if we look now in patient, this is a paper that on the left we still didn't publish. We already see that at the diagnosis in prostate tumor, even in small biopsy, we are capable to detect the presence of this myeloid-derived suppressor cell. And on the right, this we published in 2018 on a Nature paper. If we look at even a small metastatic tumor lesion in the bone or in the lymph node of patient, we see the presence of a high percentage of myeloid-derived suppressor cell. One slide more. So how does it work, and why we think that targeting this immune population could be effective for the therapy of prostate cancer patients? So once the tumor forms, as shown here on the left, the tumor starts to secrete a number of factors which can promote the expansion from the bone marrow of a patient of this myeloid-derived suppressor cell, which are highly heterogeneous. However, during the years, we were able to identify a receptor present on the membrane of this myeloid-derived suppressor cell, which is called 6ER2 receptor, which is a key receptor to allow this cell to infiltrate prostate cancer. And back in 2014, we show for the first time that a combination of an inhibitor of the 6ER2 receptor, we are now in the clinic, with this was able to enhance the efficacy of a standard of therapy in the metastatic castration-resistant prostate cancer, which is named docetaxel therapy. Therefore, proving that this, the elimination of this myeloid-derived suppressor cell from the tumor microenvironment of mice or patient could be beneficial for enhancing the efficacy of a chemotherapy. Can you go next? So what are the main research interests in my lab from that moment? So we want to identify how myeloid-derived suppressor cell can promote tumor progression in patients affected by prostate cancer. We do this by using different technology, different omic technology, single-cell technology in primary and metastatic tumor. And what we try to find is a secreted factor produced by myeloid-derived suppressor cell, which can, for instance, promote proliferation of prostate tumor cell, the migration of the metastatic capability. We also focus, as you can see on the right, identification of additional immunocheckpoint, which could be relevant in prostate cancer, and that and whose inhibition could be later on combined with therapy that block myeloid-derived suppressor cell. Next slide. So this is an example of what we have recently published. We discovered that myeloid-derived suppressor cell that infiltrate the prostate tumors in the mouse and patient can release a very high quantity of interleukin-23. This interleukin-23 can later on bind to the IL-23 receptor, which is strongly expressed in prostate tumor cell, particularly in patients affected by prostate cancer, which are treated with androgen deprivation therapy. And if you can go one slide more, what we discover is that actually the production of this IL-23 from myeloid-derived suppressor cell can activate the IL-23 receptor at the downstream pathway, thereby promoting resistance to a conventional therapy such as, for instance, enzalutamide, which is a standard of therapy in patients affected by prostate cancer, which do not respond to first line of hormonal therapy. So in the years, we have developed different therapy. For instance, now we are studying an antibody against which can block IL-23 produced by myeloid-derived suppressor cell. And if you go one slide more, what we like to do here in Bellinzona, where there is also a very active phase 1/2 clinical trial unit, we try to move this discovery from the bench to bedside. So and we have, for instance, opened during the year different clinical trial, a phase 1 trial in collaboration with AstraZeneca that it's focused around an inhibitor of 6ER2 in combination with androgen deprivation therapy, and a second trial, which is ongoing here in the Royal Matzen in collaboration with Johan de Bono, that study an antibody against IL-23 in combination with ADT. So thank you, and open to the question.
Great, thank you very much, Andrea. So we've heard from two academic centers of excellence in Switzerland on bringing from research from mice model into humans. And we also heard about facilities that have been built for that. It's a very rich landscape for collaborations, partnerships, and for developing and bringing new therapies to patients that are waiting for them. For those of you who are interested in the academic centers of excellence, on the left side, you will have a chance to go after the completion of our panel to the breakout session that's called Cutting Edge Academic Collaborators, where you can meet Lana and Andrea for the Q&A session. With that, we would like to now move to the emerging scale-up companies that are used to be spin-outs many years ago, and now are in the process of globalizing and building partnerships. And the first one, I'd like to introduce Marija Plodnek. Thank you for joining us from the US. You traveled very recently, so you must be on the jet lag, but please look forward to hearing all about Artidis.
Brian, good afternoon and good morning, everybody. From Houston. So I would like to present developments done by our company called Artidis. We are actually a spin-out from the University of Basel in Switzerland. And I joined Artidis from the university as a CEO and co-founder in 2017. where we kind of brought our scientific discoveries in the field of investigating cell interactions with tumor microenvironment using physical tools. And I will talk about a little bit how that relates to immunotherapy today. Our startup in Basel, we were 5 members at the time, and now 5 years later, almost 5 years later, we grew to 32 full-time employees in Basel, office in Houston, and also we have a strong team in Barcelona, and we will soon be expanding in other European countries as well. So the slides are going a bit too fast, so I would like to go back to the first slide a little bit. So what is Artidis all about? Our nanotechnology platform has been developed to actually for cancer diagnostics and treatment optimization, with, as I mentioned, focusing on measuring physical interactions between cells and microenvironment. And Andrea just now made a little nice introduction how important tumor microenvironment is, in particular in context to responding to different types of immunotherapies. Our technology really allows, beyond what we see with different omics approaches, how those actually translate in phenotypes, meaning what does a certain protein expression make up within the cell will make this cell more deformable, less deformable. Will these cells be able to migrate and diffuse within different tissues? Because as we know, in particular in the context of immunotherapy and the immune tumor microenvironment, one of the large issues that we have, even when we have the immune cells present in the surrounding of cancer cells or in the nearby stroma, often the issue is actually get them, and this is what we call immune excluded tumors, to come into contact with cancer cells. And that is hindered with the stroma, and understanding the physical capabilities of that stroma and then changing it are very important to improve the response rates, which despite lots of successes are still not where we would like to see them be. Also very important in that context is to actually combine different types of biomarkers. I think in the course of the panel, there will be discussion, and in immunotherapy development, there is a number of tissue-based, immunohistochemistry, blood-based, genomic biomarkers, but we are lacking understanding of those physical interactions between the tumor and the microenvironment that will allow better responses. And this is something that we focus at Artidis in developing our technology and moving forward. And more importantly, once we measure those characteristics of tumor and its microenvironment, how can we act to change it using different modalities, for example, radiation. So now we can go to the next slide. So how does Artidis actually work? It utilizes a technology called atomic force microscopy, which, funny enough, many years ago was actually developed in Switzerland at IBM. And the idea was to use it not for the applications we are presenting today, but to measure some very basic physical forces between different atoms, and typically using it in material science. Meanwhile, the technology has moved much more into biology application, and I'm glad to say that we could pioneer this technology to actually assess living tissues in their native environment and measuring those physical characteristics between the different types of cells and the matrix scaffold around them at the molecular resolution. At the heart of our technology is this probe that is illustrated on the screen. This is a silicon nitride sensor that literally works like palpation. So you touch the tissue, and by measuring the response, the stiffness, what we call response from different tissue components, we can really at subcellular resolution discriminate different types of cells, being different types of cancer cells, different types of immune cells, and then characteristics of those matrix stromal components. How our technology is currently being tested in clinical trials and where we aim to apply it is really to guide and maximize the response of solid tumors, with the focus on solid tumors to chemoimmuno and cell therapy. Interesting, this technology can be easily incorporated into routine clinical setting. So next slide, please. And it consists of a device that can at the patient bedside, so in the radiation oncology suite or in the surgery suite where the biopsies are typically taken, can immediately assess and measure the sample. One very important aspect is that it's much more than a device. It's also a platform, and this is also very important in context of understanding immune response and immunotherapy. It allows to integrate different type of patient data, clinical information, previous anamnesis, and then other types of biomarker tests that will be done either prior or at the same time as Artidis measurements. And then by integrating those, we are really getting a holistic picture in understanding what is happening with that tumor, and then the next, how we can actually address it by either using something via endogenous immune response or synthetic, what was the previous discussion, for example, adoptive cell therapies. How can we do this treatment optimization as a next step? Because the goal is not just to say, yes, this tumor is responding well or not responding. We have a number of biomarkers already available that are able to tell us something about those characteristics of tumor. But the question is, how can we then change this tumor in a way to be more sensitive, more responsive to immunotherapy? And one modality that we are using, it's combining Artidis with different modules of radiation. So we can go to the next slide. And this is, as an example here, illustrated what we call a radioscopal effect, where you would combine a high dose radiation to target the tumor and then low dose to actually affect this tumor microenvironment and to perturb those physical and biochemical interactions in a way that the tumors become more responsive, and also that the T cells migrate to the site of the cancer cells and interact and pretty much destroy the cancer cells. This is important when we think about the clinical applications, because for us, the ultimate goal obviously is to bring it to the clinics. We have already done a large clinical study in Switzerland, which was in a different context, using Artidis technology for diagnosing and predicting response to neoadjuvant therapy in breast cancer. This was a 545 in total patient study. And now we are moving forward to other applications as illustrated here. And immunotherapy response is one of them, where we are really focusing on understanding how the signatures of responding tumors versus resistant tumors look like, what is happening when we try to alter these tumors on modalities such as radiation. Do we restore? How does the signature look like when we restore sensitivity versus when we are not able to restore sensitivity? And I'm looking forward to see how the next results will look like, but we have already done a proof of concept study. We have a strong collaboration with Barcelona and also with Texas Medical Center. And this study that, for the sake of short time, I won't go too much in detail, we have done with MD Anderson, where we really combine Artidis with evaluating biopsies, therapy, and then on therapy, first using radiation and then anti-PD-1. Similarly, we are using the same modality in changing the microenvironment of tumors and then combining it with CAR-T therapies. And in the next months, we expect a number of clinical trials and preclinical studies that will, on one hand, allow us to better understand what is happening within the tumor microenvironment at the naive stage and then on treatment. And for that aspect, I think it's also very important to mention that serial biopsies is something, or evaluating tumors from solid cancer patients prior to treatment and then on different stages of treatment is really truly essential. And we are building this technology to make that possible in the easiest way, in order that long-term the aim is to, in real time, measure, adapt it, or alter the tissue in a way that we can accordingly adapt the therapy and the response. We had very positive data that show that we are able to do that, and I think the ultimate goal is to really, for those patients at first hand, they don't respond well, to improve their responses, and long term to actually expand the applications of immunotherapy. And I don't know, do I have still some time, or?
I think we're really over time. Thank you very much, Maria.
Yeah,
We'll continue this discussion in the breakout session, but first we have one more presentation on application of precision medicine within immunotherapy. I will be presenting that as chairman of Novogenics, which is a company based in Lausanne, Switzerland. Novogenics focused on leveraging immunotranscriptomics, essentially measuring gene expression profile of immune system to bring insights into how the immune system is functioning in different therapeutic setting. Whether you isolate the immune cells or whether you do it in whole blood in form of liquid biopsy, or whether you take a tissue biopsy and you extract the RNA, that data is generated on a next generation sequencing, and with algorithms and data analytics, we're able to generate reports that allows a patient or a physician or a clinician to get insights of how the immune system is functioning. This platform has been set up with over 10 years of research, which was then translated into a professional platform with application both for biopharma that are today utilizing the platform to bring insights into the immune system within their drug development pipeline, as well as into clinical research environment, where we're able to leverage this to bring insights into what the immune system is doing within different indications, including anti-PD-1 therapy, which we'll see later, as well as then for academic research in terms of advancing the knowledge base in terms of how the immune system is functioning. So in one tube of blood, in a liquid biopsy, we have about 4 million of T cells, B cells, neutrophils, monocytes, and we're able to, across the full immunotranscriptome of 16,000 genes, measure small changes, up or down regulation of these genes that are then translated into biological pathways, and what does that mean actually for the functioning of the immune system in the patient. Just to show you an example of how that works, in the case of, for example, colorectal cancer, we've completed multi-center clinical trials here in Switzerland and in Korea, where we're taking healthy individuals and also individuals with colorectal cancer, all of whom have gone through colonoscopy, so we know exactly what stage of disease they are in, and we can map essentially in their immune system how as a healthy individual goes into development of advanced adenoma, we get IL-23 mediated signaling. And as the disease progresses and we go into stage 1 and 2, early stages of the disease, we get complement systems, IL-17 mediated signaling, oxidative stress, angiogenesis. And finally, as we go into later stages of the disease, stage 3 and 4, where the individual becomes symptomatic, we get neutrophil and platelet degranulation. So this sort of gene expression profile evolution is used to identify patients early. For example, we currently have a product on the Swiss market with now Health Center insurance coverage, which allows with a blood test to be able to detect which individuals are in the process of transitioning from healthy to advanced adenoma and early stages of cancer. Now, within the anti-PD-1 therapy of immunotherapy, as we know, for example, in bladder cancer patients, there is a very low response rate of about 20%. What we have shown is this system can do is identify molecular pathways that are up and down regulated in these patients that are predictive of the responders versus non-responders, to the point where we can double the response rate by doing a patient stratification strategy. This has been presented as ESMO, and we finally received a consortium commitment from the European Union to advance this into a product to bring it into the market. We are grateful to work with a large number of academic and professional centers here in Switzerland and abroad, and we are very much benefited from the rich ecosystem here in Switzerland, both from the academic center as well as from the industry.